Hey everybody, welcome to the final exam review. So we will go through a PowerPoint that I literally just emailed out. I apologize that you're just getting it in your email through the course, but it is there. So I will be recording this, of course, putting the lecture record or the review recording up in the announcements. And all you guys have this week is to take the final exam and to do your lab practical. So make sure that you know when you're going to be able to take the final exam. Has to be done by 5 o'clock on Friday. And all other coursework, if you're missing anything, which I don't think anybody really is, that has to be submitted by midnight on Friday. Now, remember, with the Micro 2 final exam, it's going to cover both Microbiology 1 and Microbiology 2. So it will be comprehensive over both. Same with the lab practical, it could be comprehensive over both. The nice thing about the lab practical is you've been doing a lot of unknowns anyway, so hopefully that's kept some of the practice up on both Micro 1 organisms and Micro 2. So with that being said, the other big thing to remember on these exams is both the final exam and the lab practical have to be passed with a 73% or higher on each exam. So you got to get a 73% or higher to pass the exam and to pass the course. So if you do not get higher than a 73% on either one, you will be allowed one retake opportunity, and that must be done by Monday. And you have to work with your MLT program coordinator on when on Monday they want you in to do that. So I have a retake on the regular final exam in case you don't pass that, and then I have a retake on the lab practical that you would do on Monday if you don't pass that. They are different tests. They're not the exact same test again. So they're similar, but they're different. So that's that. I will say this, from all the years I've taught micro, I don't have that many people not pass micro to the final exam and the lab practical. By this point, I think we're strong, and I feel like you guys will be good. You guys are a strong, strong group. So I'm not worried about anybody in here at all. So just remember, go into with confidence. You guys know your stuff by now. We've drilled it down, and just think how exciting you guys, I'm assuming almost all of you are done and going to practicum. Some of you might have a different schedule, but for those that are done going to practicum, this is really exciting. You're finally getting to that very end of the road. So with that being said, let's jump in. The exam itself, before I share the screen, is going to be 110 points for the written exam. And so there's basically 106 questions. The majority are multiple choice questions. I think there's one essay. I believe all the others are multiple choice, but there is at least one essay, but overall 110 points. So now that we've said that, we are going to share my screen. I just had to do something real quick, so I apologize. All right. So I'm going to bring up, first I'm going to just show this. I did not send this to you just because, I don't know, it's nothing really on here, but I'm going to go through it real quick, but I didn't send it to you. So you're not going to find this in your email. But essentially, it's just going to say the key areas to study for the final exam. And so again, all through Micro 1, Micro 2, it's basically everything we've talked about. It's all the different augurs. So we are going to go through that today on the other PowerPoint. We'll definitely go through all the augurs again, what categories there are, what they fit in, some specialized augurs, that kind of thing. Antibiotics. That I don't review today because I find that is all memorization. So go back to your antibiotic study guide, go back to your antibiotic PowerPoint, whatever it might be that you have, and relook at the categories, what the mode of action is, meaning does it target just gram-positive or does it target both gram-positive and neg? Also know what it basically inhibits. Does it inhibit DNA synthesis? Does it inhibit protein synthesis by 30S ribosomes? Does it inhibit cell wall synthesis? That kind of thing. So that's what you need to know, antibiotics. And then also be familiar and confident with the term minimum inhibitory concentration, MIC. So that comes out of that area as well. So go back and relook at that. Again, it's just a lot of memorizing on that stuff. There's not much to make it fun. Stains, we will review stains today. Of course, we should be experts at the gram stain by now. We'll also review acifast, acridine orange, ormine, rhodamine, and calcifluor white. So those are your stains. Make sure you're familiar with your TSI and IMVIC reactions. You should have PowerPoints specifically on each of those and memorize those reactions for the Enterobacteriaceae bacteria that we just learned. I'm sorry, my phone is ringing. right, sorry about that. Of course, somebody calls when you don't want them to. All right. So make sure you review your TSI and MVIC reactions. There's those 2 PowerPoints, and just making, you know, we definitely have had questions like that before where it gives you the TSI reaction, and then you get 4 choices. So, you know, kind of go back through that and rememorize all of that. All right, also other tests. We need to know catalase, coagulase, and oxidase, indole, the reagents involved, basically what the reagents are, what they're kind of looking for. So for example, knowing that indole is looking for that tryptophan, that kind of thing. So just know a little bit about the color reaction that they take or what reaction they take, like oxidase turns purple when positive. Coagulase, we're looking for agglutination, and there's free and bound coagulase, that kind of thing. Catalase is hydrogen peroxide used. So that's that. Of course, parasites and mycology are big. We need to definitely know the pictures for the lab practical. You will be given pictures to identify on the lab practical. That's a big part of it, and that will be of the parasites and mycology. So hopefully you've been using those pictures with the key that goes with them to practice. I sent them out not too long ago again. So really know that, especially for lab practical, but also know about the bigger, the different questions I always like to ask on parasites and mycology from previous. So relook at that information. You can cross off viruses. We used to have viruses as part of microbiology. We took it out when I remade it, so you actually don't need to worry about this anymore. And then, of course, anaerobes. You had a week on anaerobes this session. Mycobacteria we had a week on. Spirochetes, mycoplasma, intercellular. I think that was in week 10 of microbiology 1. So go back, relook at that material. Knowing the 3 categories of toxins, we just did that a couple weeks ago, so that's not very long ago. And of course, all of the bacteria that we've discussed, there are different key clinical significance, basically the disease and their key characteristics. So, okay, so basically everything from micro 1 and micro 2. All right, so you get the gist. Now let's jump into the PowerPoints I did send you just to help review some of this. I'm not going to go through all micro 1 and micro 2. We'd be here forever. So essentially I'm going to go more through the micro 1 stuff just because that's been the stuff that we haven't looked at in a long time. The stuff for micro 2 this quarter, I'm not going to go back through because that was recent. So you need to go back and look at that stuff for yourself. So first we're going to start with terms. We should be very familiar with all of these terms. We should know that normal flora is basically the bacteria that resides in us or on us. Normally, that it's there for a good purpose. It's there to help with our immune system. Colonization is the keywords on this one are always persistent survival. It's the persistent survival of microbes on the surface. Opportunist is exactly how it sounds. They're waiting for an opportunity, an advantage to cause disease. So they're kind of waiting for maybe your immune system to be down or something like that for them to flare up and cause disease. Virulence, lots of bacteria have different factors that they can produce and have that helps them cause disease. The more virulent, the better at disease they are. And then pathogen just means disease causing. Nosocomial is hospital-acquired infection. Can't write. And of course, fastidious is picky little buggers. They need extra nutrients. So this first slide should be easy for you guys at this point. All right, now we'll start with our stains because that was one of the very first things we started with back in Micro 1. Gram stain, you should definitely be able to list out 4 reagents for me right now. I'm not going to call on anybody in the room right now. I'm just going to write these out. But you should be able to write out, let's see, I'm just going to go right here. The first one is crystal violet. That's considered the primary stain. Then you have iodine, which is the mordant, which helps bind the crystal violet down into the cell wall. Then you have decolorizer, and finally you have safranin, which is your counter stain. So those are your 4 reagents of gram stain. The other question I ask is, at which step can we differentiate a gram pos from a gram negative? I'm just going to bring this back up here. And so the step that you can differentiate gram pos from a gram negative is, again, the decolorizer step. So that's when you can first tell them apart. And what makes a gram pos different from a gram negative? So a gram pos has thicker peptidoglycan, and teichoic acid cross-links. So with that thicker peptidoglycan layer and the teichoic acid cross-links, that together helps embed that crystal violet stain into the wall and not rinse it away. All right, as far as acid fast goes, there are 3 reagents involved there. I'm just going to write them. I think I have room. No, stop. Here, I think I have room right here. Okay, so you have your carbofuchsin, which is your primary stain here, your acid decolorizer, and your methylene blue, which is your counter stain. So again, if it is acid fast positive, it'll keep this purple red color that this is, and if it's acid fast negative, it will be blue. So acid fast positive are purple or red, whichever one you want to call it, and acid fast negative is blue. All right, as far as the methods, remember there's both the Ziehl-Neelsen and the Kenyon methods. We just went back through all of this with the mycobacteria chapter, so this should be very familiar to you. Again, the difference is Ziehl-Neelsen involves heat or boiling, and Kenyon does not. So you should know that already from mycobacteria chapter. As far as what is the stain targeting, these cell walls are different. They are made of mycolic acid instead. Okay, so that is that slide. Other little fluorescent stains. So all 3 of these are fluorescent stains. They all target something different. Ormine, rhodamine targets mycolic acids. So this would be a fabulous choice to use on mycobacteria or anything that has mycolic acids, just like what we use the acid fast stain for. Acridine orange binds nucleic acids, and calcifluor white binds fungal cell walls. Okay, so that's that with the fluorescent techniques. Now onto our augers. So remember, we have 4 auger categories, and we're just going to quickly redefine them. So supportive basically means grows almost anything, and we always say almost because it will not grow fastidious because those are extra picky. Enrichment has additional nutrients. It's been enriched, and this will grow fastidious because it has extra stuff for them. Selective will specifically grow one group of bacteria, so either gram-pos or gram-negatives. It's just going to grow one group and inhibit the others. And differential is going to basically categorize the bacteria growing based on the characteristics that they have. So is there fermenters versus non-fermenters, for example. So those are your basic 4 definitions of the categories. So now we're going to get into each of our augers and categorize them together. So we're going to start with blood agar plate because that is the most common agar we use. It has 2 categories. It is going to be supportive because it basically grows pretty much everything. That's why we use it for everything. And it is also differential. And the reason it's differential is based on the hemolysis patterns that can be seen on the agar plate. So again, you have alpha hemolysis, which is a partial lysis that's more green in color. You have beta, which is a clear lysis, and it looks, well, it is clear. It's a complete, I should say, lysis, and it looks clear halo-like around the colony, and then gamma, which is nothing. So there is your hemolysis, and that's why it's differential. All right, chocolate agar is supportive and enrichment. And basically, it grows everything as a result, including the fastidious. And so that's kind of it there. So, yep. All right, thioglycolate broth. This is the one that's in a broth or a tube, so it's liquid. And this is supportive, and it's also enrichment. So again, everything. The other part with the thiobroth is, remember, sometimes there can be little patterns inside the thiobroth tube based on what it's growing. Now, they're not always picture perfect, but sometimes it happens. So if you were to see like cloudy, round spheres or balls, that would be suggestive of something growing a gram-pos cocci. If it is diffuse, meaning cloudy throughout, that's more of a gram-neg rod. If it is only at the top of the bottle, that is a strict aerobe, meaning it had to have oxygen. And if it's cloudy only at the bottom, that is an anaerobe trying to get away from the oxygen. So those are some of the patterns that the thioglycolate broth can appear. All right, CNA agar, hopefully you guys have said it's selective, and it is selective because it grows gram-positives. And then PEA, same thing. So labs are going to have one or the other. So gram-positives only. So that's it on the first chart of agars for the categories and what they do. We will go to the next one. If you weren't done writing, remember I'm putting the recording up so you can just go back to the recording and pause it and write down everything. All right, McConkey agar, very common agar used. This is selective because it's growing gram-negatives, so specifically gram-neg rods, I should say, so GNR. And it is also differential, and it's differential based on lactose fermentation. There'll be purple colonies, and non-lactose fermenters are clear or colorless colonies. Hectoenteric HE agar, same category, selective and differential. I cannot write. Oh, there we go. Again, they are specifically looking for Salmonella and Shigella, which are, of course, both gram-neg rods. And again, differential based on fermentation or not. And these two are known to be non-fermenters, so they are going to appear blue-green on the agar when they grow. So Salmonella and Shigella are present, there will be a blue-green color on that agar. And also, because Salmonella is well known to be an H2S producer, remember it will get that black center to it. XLD is just like HE, so labs would have one or the other, so it's selective differential because, again, growing Salmonella and Shigella. And again, these are known non-fermenters. And on this agar, they are going to be colorless when they grow on this agar. All right, Syn agar is selective. And this is specifically growing Yersinia and really wanting to look for Yersinia enterocolitica. Yeah, I spelled it right. And remember, that looks like bullseye colonies when they grow on Syn agar. TCBS is selective, and this is specifically looking for Vibrio. So it will grow the Vibrio species, and if it's Vibrio cholera, that will be yellow colonies. The other Vibrios, like Parahemolyticus and Vulnificus, tend to be green, I want to say. Yeah, it's green. So, okay, there's that one set. All right, we have our Martins. Their Martin is selective, and it is specifically looking to grow Neisseria. And because Neisseria is fastidious and picky, it needs to be enrichment. So that way Neisseria will grow. Now this has a ton of antibiotics in it to make sure that only Neisseria grows and nothing else. So we need to know those. So we have colistin, which inhibits gram-negative rods from growing. It has vancomycin, which inhibits gram-poscoxy. We have trimethoprim for inhibiting Proteus. This is getting to be long. And then we have nystatin to inhibit yeast. So there's your antibiotics found in there. And Martin Lewis is selective and enrichment because, again, it's looking to grow Neisseria. Very similar. The only difference with antibiotics is that it has more vancomycin and it has mycin to inhibit yeast. Instead of nystatin, it uses mycin, so that's the difference. All right, Bourdet Ginkgo is selectively growing Bordetella pertussis. Bourdet, Bordetella, I mean, the words are almost identical, so that should help. Lowenstein Jennings is selective for growing mycobacteria. So we just mentioned that one not long ago. The other one that's used commonly for mycobacteria is Middlebrook. It's not, well, I shouldn't say it's used commonly anymore. I don't think they use it a lot, but the other agar that we mentioned, so for mycobacteria, it would be Lowenstein Jennings and Middlebrook. I should also mention back to the other one on Bordetella, the other one we mentioned here was Reagan Low for Bordetella. And then BCYE is buffered charcoal yeast extracts. So this is selectively looking for Legionella. Okay, so that is those augers. We pretty much have hit the majority of the commonly used augers and then some of the extra specialized ones. As we go, if there's other specialized augers, I'll be sure to try to mention them as we keep going. All right, so now let's discuss our Gram-Pos cocci. So that was the very first group of bacteria that we learned in Microbiology 1. And so one of the first things we realized is out of the two major Gram-Pos cocci group, we have staph and strep. We had to differentiate them with a test. And the very first test that helps differentiate these is the catalase test. So you should definitely know by this point that staph is always catalase positive and strep is always catalase negative. So that's how it separates them. And again, the reagent used is hydrogen, hydrogen peroxide, H2O2 is the reagent. So bubbling or no bubbling, very simple test. All right, so starting with our staph, this was a short list of the staphs that I definitely need you to remember and know. So colony morphology, Staph aureus, again, let me first say in general, staphs tend to be medium or larger than strep. They tend to be medium-sized colonies, creamier. Staph aureus tends to be beta hemolytic, and it sometimes has a yellow hue to it. Again, remember that aureus means gold. Gold is yellow, so that kind of helps put it in perspective why they named it that. Again, lab test results, we would, as soon as we see it's a Gram-Pos cocci, we would catalase test it, should be catalase pos. The minute that we know it's catalase positive, we are going to coagulate test it. And Staph aureus is hugely coagulase positive. Remember, there is a free and bound coagulase test that you can perform. There's both. Sometimes labs will do both of them, one to confirm the other. A lot of labs will only run one of them. They won't always run two of them. So just remember that and take a look at that back out of your chapter. As far as disease, it's just so much. It causes so many different things, UTIs, wound infections, different skin infections, hair follicle infections, toxic shock syndrome. I mean, the list goes on and on and on. So you can't even pinpoint it here. It's just always a pathogen. Extra stuff here to mention, it is found as normal flora in our nares, our perennial area. So there is some normal flora here. And then the other thing is to not forget MRSA, methicillin-resistant Staph aureus. You would treat that strain with vancomycin instead. And I think the only other mention I want to say is there is a special auger to screen for Staph aureus called mannitol salt auger, MSA. So I think we're covered with Staph aureus then. I'm going to come back to it and add a couple things in a minute. All right, Staph epidermidis is gamma hemolytic, so basically non-hemolytic, and it tends to be white. You could call it gray as well. I think it's white usually. It is catalase positive because it is a staph, and it is coagulase negative. Disease, typically Staph epidermidis is contamination because it is huge normal flora of your skin. That's why it's called epidermidis. But for that, those times that it does cause disease is with medical devices. So one of the virulence factors it does have is to produce this slime and make like a biofilm environment, which can really cling on to like prosthetic pieces or medical devices, anything that's going to go in the body. And if it clings on and it gets introduced into the body in a sterile area, then it will cause infection. Staph saprophyticus looks just like Staph epidermidis, gamma hemolytic, white, gray. All right, it is catalase positive, coagulase negative, and the big disease here, you should all know this, UTIs in young women. And again, it is found as normal flora of the genitourinary tract. So upon sexual activity, it can get introduced in, especially if they don't clean themselves well. So the big thing becomes differentiating epidermidis from saprophyticus because they look alike, so far they're testing alike. One tends to be more contamination for the most part, the other does definitely link with UTIs. So we have that novabiocin antibiotic disc. And again, Staph epidermidis is susceptible to that disc, whereas Staph saprophyticus is resistant. So that will help separate them. Okay, Staph intermedius. A lot of times it does look very similar to the rest of this. So, we're just going to leave that. It is catalase positive. This is the one that can be coagulase variable. A lot of times it is negative, coagulase negative, but there are a few strains that are coagulase positive. So if that's the case, you can do extra tests to help separate it from Staph aureus. So if you PYR tested it, it would be PYR positive. If you Vogt-Krosker tested it, it would be VP negative. And so if we go up to Staph aureus, that's PYR negative and Vogt-Krosker positive. So you can see how those two tests separate them back out. Disease, we linked this one with dog bite infections, but it can probably pop up in a few other things. And finally, micrococcus can really take on any color. It's been known to be orange, pink, white, tan, and so on. So it really can show up kind of almost any color. It's grouped here because it is catalase positive, so it does fit well as a Gram-Pos cocci catalase pos. It is coagulase negative, and if you were to oxidase test it, which you really shouldn't, but if you were, it would be oxidase pos. And it doesn't really cause disease. It's pretty much always contamination. We always mention it, though, because you will hear of it once in a while as being contaminants. So that is your Staph Gram-Pos cocci right there. That's very doable for you guys. Okay, let's go on to strep, which is our other big group of Gram-Pos cocci. So remember, streps, some of them are grouped together based on their Lancefield group, which are their carbohydrate antigens on their cell wall. So there is we'll mention the group name for two of them here, so based on that Lancefield group. The other big thing with streps is hemolysis. When we know we have a Gram-Pos cocci and we catalase test it and it's catalase negative, the very next thing you do is look at what hemolysis is it to decide the next test that you're going to run. Okay, so we're going to start with strep pyogenes, which again, you should already know is named Group A strep. So with hemolysis being big, it is beta hemolytic. Now remember, streps altogether are tending to be pretty small. They're small colonies, so I'm not going to keep writing that. I'll just write the hemolysis. So it is catalase negative because it's a strep. Once we know it's catalase negative, beta hemolytic, the first thing we want to run is a bacitracin A disc, and it is susceptible to this. There. So that is susceptible to that bacitracin A disc. If you did a PYR test here, that would be positive. If we did CAMP test, that would be negative. And the other one finally is hippurate hydrolysis, which that would be negative. All right, disease, so much. Strep pyogenes is always causing disease. It's a huge pathogen. It is never normal flora. It is always a carried by somebody potentially, but it's never normal flora. So there's so many diseases. Remember the scarlet fever, strep throat, of course, which is called pharyngitis. Impetigo, so a lot of skin infections, toxic shock syndrome, necrotizing fasciitis, which is that flesh-eating disease. And there's so many more. Remember, the other big thing with strep pyogenes is there were two post-strepto, and now I'm writing down below if you can see, two post-streptococcal infections or diseases that occur, so after having a strep pyogenes infection, and that would be rheumatic fever and acute glomerulonephritis. So keep those two in mind as well. All right. Going on to strep agalactiae, this is group B strep, and this is also beta hemolytic. So we're going to run the exact same test. Catalase negative, bacitracin A disc resistant, PYR negative, CAMP positive, and hippurate positive. So right there you can see how you separate group B from group A. Disease, the only thing I asked you to remember was neonatal meningitis. So this is the one that sometimes pregnant women carry in their vaginal tract. So at some point during their pregnancy, we swab, see if they're carrying this. If they are, they get antibiotics to wipe it out before they give birth to help prevent passing it to the baby and risking the baby for neonatal meningitis. So I've always said, think group B for baby. Strep pneumoniae has no group name, so don't worry about a group name. And this is alpha hemolytic. So test results, it's catalase negative, of course, it's a strep. And in this case, we're going to run the Optekin P disc, and this would be Optekin susceptible. You could also run a biosolubility test, and this would be biosolubility positive. Disease, of course, it's the #1 cause of pneumonia. It's named for it, and it also causes meningitis. So there is that vaccine for older adults with meningitis, to prevent meningitis, I should say. So 2 very big diseases there. And viridin strep, these are a bunch of alpha hemolytic streps that are just grouped together under the name viridins. Catalase negative, Optekin resistant, and biosolubility negative. So you can see how that separates the 2 apart. When these do cause disease, they kind of cause endocarditis and dental problems, dental infections, essentially. Some common examples of ones that fit into this category is strep mutans, strep mitis. Those are both viridins, alpha hemolytic streps. And finally, Enterococcus. Enterococcus, again, is a gram-positive cocci. It fits best with these because it tends to be catalase weak positive. It might be so weakly positive that you miss it and call it negative. For hemolysis, this can really be any hemolysis. So in this case, ideally the test to run with Enterococcus would be that it loves to grow in the presence of salt. So if you put it in a salt broth, like 6.5% NaCl, that would be positive. It is bioesculin positive and biosolubility negative. So that will help separate it from anything else. As far as diseases goes, it's associated a lot with nosocomial hospital-acquired infections. Okay, so that's it for Enterococcus. So overall, that's all your streps, your gram-positive cocci. Oh, you know what? The only thing I forgot to mention is strep pneumonia, remember, is a diplococci. It occurs in pairs said to be shaped like cat eyes or lancets. So I forgot that. Yeah, strep pneumonia is pairs, diplococci. All right, that is it on streps. So for the next gram-positive group, we have gram-positive rods. So we had a whole PowerPoint on this. I have them separated in 2 slides here. So we're not going to probably get as detailed as we were with the gram-positive. I'm just going to hit the high points of what you definitely should know on these little guys. So we're going to start with our bacillus. There's just 2 bacillus organisms that you need to know. The rest of the bacillus species is pretty non-pathogenic. Tends to be a lot of normal flora of our skin and other stuff, so we don't really see it except for sometimes contamination. But we definitely need to know about Bacillus anthracis and then some on Bacillus cereus. So as far as colony morphology goes, for Bacillus anthracis, they're very large colonies. These are the ones that are said to be Medusa head-shaped or comma-like projections. So they're not nice and round circles. And they also tend to be a little sticky when you kind of try to go pick them up with the loop. As far as lab tests go, they are catalase positive. These are non-modal. Oh, I should also add it's non-hemolytic. I forgot that. All right, non-modal, penicillin, susceptible, and lecithinase positive, which we didn't talk a lot about that. It's just an enzyme that it possesses, but I'll put it there. All right, for disease, of course, anthrax. Remember, there are 3 forms of anthrax. There's the GI, the cutaneous skin form, and the lung, the inhalation. So again, the lung inhalation, if it's infecting their lungs, that's always bioterrorism. Skin is probably the most common to see it. So remember, you can find Bacillus anthracis in the soil. And so if it gets picked up by an animal and you're handling the animal and get it through an abrasion and it affects your skin, that's most commonly what we're going to see. So based on that whole scenario, that's where they named it wool sorters disease, from shearing the sheep and picking it up out of the wool. All right, Bacillus cereus is large colonies as well, but they call these more spreading. They don't have those projections or anything. And then is beta hemolytic. I cannot spell. Oh my goodness. Okay. All right. Catalase positive. Okay. These are motile and penicillin resistant and lecithinase positive. So you can see how the motility in the penicillin result helps separate between anthrax and cereus. Also, the hemolysis is a nice factor there. For disease for Bacillus cereus, I said two things: food poisoning and eye infections. So I think we saw mention of both of these in our last few chapters. The GI chapter, we definitely talked about that Bacillus cereus does possess a neurotoxin for food poisoning, and then we also mentioned it in the eye chapter for eye infection. So it got re-mentioned in both. As far as extra, the only thing I forgot to mention is on both Bacillus anthracis and Bacillus cereus, these are very large Gram-Pos rods, spore forming. So don't forget, they are spore forming. So I'm going to put that up in both. So large Gram-Pos rods. They're said to be the ones that look like boxcars off of a train. So they're very in your face. All right, Cranibacteria diphtheriae. This is the one that has some unique features on the gram stain. I'm going to put that over here first before I forget. Again, they are Gram-Pos rods, but they're said to have the Chinese letters look to it, or they can take the shape of the letters V or L, or they're said to be palisading, which means lining up side by side each other. So that's kind of unique on the gram stain. As far as colony morphology, there are some very specialized augers that are used to grow this. If you grow it out of cysteine tellurite, they would be brown, black colonies. They also can be beta hemolytic on blood auger. So nothing much else there that you need to remember. Lab test-wise, it is catalase positive. The big test here is growing it out on Tinsdale auger and having a what we call brown halo. And disease, of course, this is diphtheria. So where you get the membrane on the back of the throat. So that's the classic sign of diphtheria. Listeria monocytogenes tends to be smaller, beta hemolytic. As far as lab test results goes, it is catalase positive. It has huge motility. This is the one with the tumbling motility, such a big characteristic of Listeria monocytogenes. Or if you were to do a different type of motility test, it would take that umbrella shape. Either way, both are very classic to Listeria monocytogenes. This is also CAMP positive. Okay, so for disease, neonatal meningitis, this is the whole one with pregnant women need to be careful on what they're eating. They shouldn't eat certain deli meats, which I never knew when I was pregnant. I guess I didn't realize that, so I'm good, but I had no idea. But they can't eat certain deli meats. They can't eat certain cheeses that are homemade or softer, that kind of thing. So there's just some foods that you're supposed to stay away from. And that should be it. So that's it for your Gram-Pos rods on that chart. So let's do the other Gram-Pos rods here. This stuff gets to be even less. We don't need to know a ton of these ones. For erysipelas rosea, what it is, I'm not really worried about the colony morphology here. So, you know, you can list just gray, whatever. But as far as lab test results go, it is catalase negative. And this is the one that is H2S positive. It is the only gram-positive rod that produces H2S. So that's always a very key result with it. Disease-wise goes, it produces erysipeloids, which are basically skin ulcers. It's a skin infection. So, and it comes from handling animals. So this is definitely carried by animals like pigs. So, and upon handling them, you can get that erysipeloid skin ulcer. All right, Nocardia. This one is branching, so it can sometimes have aerial hyphae. Is catalase, it's actually catalase positive, but this is partially acid-fast. So that's a unique feature in that you can use acid-fast stain for it. And disease-wise goes, it causes a few things like skin infection, a pulmonary infection, so I'm just going to leave it as that. Gardnerella vaginalis, I don't really care about the colony morphology. It is catalase negative. This is the one, though, that what you really need to remember is the clue cells. So, again, a clue cell is basically a squamous epithelial cell filled with the Gardnerella bacteria. And so you find this in bacterial vaginosis. So it's named for kind of what it does. So if somebody were to have a vaginal bacterial infection with Gardnerella vaginalis, if you were to get their wet prep, so if a physician swabbed their vaginal tract to send it to us to look at it at a microscope, or we could also see it in urine from them, you are going to see these clue cells. So they are those squamous epithelia filled with the bacteria. And you can always see that it's them because the outline of the cell gets really blurry looking, and then it's very refractile and, you know, kind of junky looking on the inside because of all the bacteria in there. That's definitely a key feature of Gardnerella vaginalis. And then if you wanted to grow it, there is a special agar that will grow it called Phenagar that will help grow it, because that will help suppress any of the other vaginal normal flora so that it grows out more. Lactobacillus, this one basically is a good bacteria. It's one of our ones that is in yogurt that helps with everything. So I'm just going to leave it like that. You don't really need to know much about that. And then, as I said before, making sure you know your antibiotics from Micro 1. So that's listed there. The other things that I did not list on this PowerPoint yet that I want to do, so we did talk about Haemophilus within Microbiology 1. Remember, Haemophilus is a very fastidious guy, so it's going to want to love to grow on chocolate agar. You have some major Haemophilus species. We have Haemophilus influenzae, which is a big cause of meningitis, especially if it's Haemophilus influenzae type B. There is that vaccination now for children, so it's really lessened how much that occurs in younger children, but that was something big. And then it's a huge cause of acute epiglottitis. And also the subspecies Aegypticus. So Haemophilus influenzae aegypticus causes pink eye. So those are some of the things. It can also cause other types of respiratory infections, but we're going to leave it listed like that for now. You also have parainfluenzae, again, causing lots of upper respiratory infections. Haemophilus ducreyi, which is your STD. It is causing a chancroid genital in the genital area. And then finally, Haemophilus aprophilus, which is part of your HASIC group. Let's rediscuss the HASIC group. HASIC is a group of bacteria, and I'm writing down below in the notes section now, that all cause slowly progressive endocarditis. So that is why they're grouped together. So it would be Haemophilus for H. A is actinomyces. C is Cardiobacterium. E is Eikenella. And K is Kingella. So be able to list those and state why they are grouped together. All right. The other thing with Haemophilus is they are gram-neg rods. They are very tiny gram-neg rods. So a lot of times they said they were like junk in the background. They are oxidase, well, it depends on which one you're talking about, but Haemophilus influenzae is definitely oxidase positive. The big thing with these is that they need X or V factors because they're fastidious. So let's continue this here. All right, so X and V factors. X factor was hemin, V factor was NAD. So Haemophilus influenzae needed both X and V. Haemophilus parainfluenzae, I can't type, needed just the V factor. Haemophilus ducreyi needed just the X factor. And Haemophilus aprophilus needed neither. So keep those test results in mind. The other big fastidious organisms that we talked about was Neisseria, and then Moraxella catarrhalis was in the same chapter. These two, again, are fastidious, so they're only going to want to grow in chocolate agar, and they are gram-negative diplococci. So they occur in pairs. They're little negative cocci in pairs. They're said to be kidney bean shaped. So the major organisms here is Neisseria meningitidis, which again causes meningitis. Nice, that is the name for it. And then Neisseria gonorrhea, which causes gonorrhea. Love it when they're named for what they do. Moraxella catarrhalis is a huge cause of upper respiratory infections. We saw that listed so many times in those few chapters that we just went through, like on eyes, ears, sinuses, upper respiratory. We saw it listed in all of those. All right, as far as test results go, these are oxidase positive. And then the big thing with Neisseria is sugar utilization. So again, Neisseria gonorrhea will only ferment glucose. Neisseria meningitidis will ferment glucose and maltose. Moraxella, I cannot spell that. None. It doesn't ferment any of those sugars, but it is DNase positive. And we talked about that originally back in Micro 1. Okay, so that's just some others. There's a bunch of other fastidious organisms we mentioned, like we talked a little bit about Legionella, Francisella. So there's a bunch in that chapter PowerPoint with Haemophilus and stuff. So go back and relook at all of that. I'm not going to mention it for time's sake, but that was all part of there. So like I said before in this one, this kind of went through what we needed to know here. So essentially just go listen to any recordings from this session, go look at any PowerPoints from the two micros together. So that should be it. I think we've covered everything we need to. I'm going to stop sharing my screen for now. And so, yeah, if you guys need anything before you take the exam, just, email me or whatever you need. Otherwise, good luck. Have a fabulous finals week and have an even better break week. And keep in touch. If I don't have you or see you ever, well, not that I see you, have you again, please keep in touch. I'm on LinkedIn. Feel free to connect with me, or you guys have my email. I always love to hear how everything is going. So have a great finals week and good luck, everybody. Thank you.